This is a study of kind of subgroup phase 2 study from the large data generating triple negative moonshot project that we had at the MD Anderson about 10 years ago. So the infancy of the study was trying to understand the difference in heterogeneity of the biology of triple negative breast cancer. So we know that triple negative breast cancer is a basket of what is not, so we call it like a trash can. Like if you're not ER, if you're not HER2, then you become a triple negative breast cancers. But we all understand there's a difference among different potential subtypes. And one of the main subtype that is quite distinct from any other basal-like cancers is the LAR or luminous androgen receptor dependent subtype. So what we were trying to do is in that whole Artemis SA data set is that anybody with the stage 2 to 3 or even like a high risk stage 1 comes into our door, we characterize molecularly. Everybody gets the anthracycline-based therapy for 4 cycles, but even before 4 cycles, that if there is any prediction, these patients will more likely to not, fully respond and achieve the PCR. We have an intervention of the phase 2 arms that, based on your characterizations, you could actually be allocated into different arm. This arm specifically looking into the enzalutamide, which is a direct targeting of the AR pathways, combining them with the Paclitaxel in the second part of the neoadjuvant therapy to see if we can actually achieve a improved pathological complete response rate. So the target in that population, the baseline based on the previous studies was set more like 5% or so, and then we were hoping to improve that at least by 20 to 25%. In actual, our small cohort that we were able to complete, we actually showed the increase up to like 40% and above. So we met the primary endpoint. The tricky part there was, though, it was such a small cohort when we are trying to identify whether there is any kind of true key biomarker to see either this could actually achieve, from patients who was not going to respond to the PCR. So we have identified future, biomarkers to validate like FGFR pathways and some of the other pathways that we haven't looked into. So there's a details of discussions, and that is on the paper that is recently published. The other learning we were trying to do is that people call LAR subtype in a very heterogeneous form. Some say AR 10% and above, some say AR 30% and above, some say we have to do an RNA-seq. So we also compare the RNA calling of the LAR versus the IHC. And then if you have an IHC cutoff of 70% and above, it seems to be well matched with the LAR RNA calling. So we think that future studies could use this as a reference, and we're actually currently actively working on better strategy to improve the outcome of these populations.